Good morning. My name is Jon McCullough, and today I will be discussing our work on SuperMUC NG, which has been working towards the human scale simulation of blood flow in a virtual human with HemeLB. So firstly, I'd just like to thank the various other people who have supported this work, both in terms of funding and in terms of technical support. So the names on the left are just the start. They're variously people who have helped develop the most recent iteration of HemeLB, those who've helped with performance analysis, and those who've helped us get it running on SuperMUC NG after some particular issues or indeed in block operations. So thank you very much to them, and to those names who I have forgotten, thank you very much in lieu. So in terms of funding bodies, the development and work of HemeLB has been funded through the UK COMES project, which is a UKRI project, and the CombioMed project, which is a European Union Horizon 2020 project. So those have both funded the development and my work on HemeLB. The POP Centre of Excellence and LRZ itself for funding, for supporting our work from a more technical side of things, both in terms of code performance analysis and indeed the funding and the allocations to run on SuperMUC NG. So thank you very much to all of those. So for the rest of my talk, I will today firstly discuss what is the virtual human concept. Then I'll provide some more details about HemeLB and its place within our vision for the simulation of the virtual human. Then I'll discuss some performance results that we've obtained on SuperMUC NG using HemeLB, and as well as some challenges that will be needing to be addressed as we move towards ever larger computer scales and simulations. And I'll also provide some details of a case study that we've been working on with some results in human scale blood flow in arteries and veins. So the virtual human. So as everyone should be well aware, the human anatomy is a complex system of interacting organs and systems of nerves and blood vessels and in the current world, immune system and many other interacting factors. And all of these are unique to each individual person. And so when a patient or an individual comes in for medical treatment, be that major or minor, these treatments can be invasive in various ways and will require individual factors of that person to be considered by the doctors to best evaluate what is the best situation, the best treatment for that given individual under their specific circumstances. And so a virtual human or a digital replica of a patient that encapsulates all of these individual variations in physiology can help a clinician, we feel, by helping them to firstly investigate multiple different options for treatments for a given individual and working out ahead of time what the optimal approach would be, to treat their patients with greater confidence in that they've evaluated different options and have worked out what the best one is. Such digital models can provide information to the doctors that are not necessarily available to them with traditional analytic and diagnostic techniques. And it can also help the patients because it can help them have a clearer understanding of what's going to be happening to them. So for a more broad-ranging view of the idea of a virtual human, I'd refer you to the CombioMed website with the link provided there, and in particular the Virtual Humans film that we did a couple of years ago, which provides a much broader overview of where our work is working towards the virtual human simulation. So HemeLB is a flagship code within both UK COMES and the CombioMed project. It is a 3D lattice Boltzmann code that simulates fluid flow and has been written in C++ with MCAR parallelism. So it is designed for blood flow simulation in the complex, sparse geometries that are characteristic of vascular networks. So various studies that have been conducted previously with HemeLB include flow through stents in aneurysms, for example, magnetical particle transport, which has been used to simulate targeted drug delivery within a complex vessel shape, and also shear stress analyses from a more technical point of view in, for example, the blood vessels of an eye. And so what we're working on now in particular is the development of human scale flow models of coupled arterial and venous domains. So having a large scale HemeLB simulation conducting the flow through the arterial vessels, and similarly a second simulation doing the venous vessels, and having these coupled together. We're also developing a GPU version of the code, which has been in particular being targeted because as new HPC systems come online, heterogeneity is becoming more and more of a theme. So in order to access and get the best performance out of future machines, we need to be able to exploit the GPU capacities of these. And so here I point to some websites for HemeLB, so for those interested can get some further resources on it. So now looking towards some performance of HemeLB that has been conducted on SuperMUC-NG. And so this work was conducted in collaboration with Brian Wiley of the POP Center of Excellence. And so what this is looking at is just a single HemeLB instance where we simulate flow through the circle of Willis. So this is a vascular structure that is located in the brain, and it distributes flow coming from the heart, from the torso into the brain, and distributes it around the brain. So this test geometry that we used, it had over 10 billion lattice sites, and it is representative of a production type simulation model. So it is of similar complexity and sparsity to what we would simulate in practice for a production type job. And so this means it's not a trivial domain, and the scaling results that we have here, where we're going from 18 compute nodes on SuperMUC-NG all the way up to the full machine scale, are representative of production capacity and capability. And so this is a strong scaling test. So the speed up that we're seeing here being nearly linear strong scaling up to the full machine scale from just over 860 nodes to almost 310,000 nodes is a very good result. And so there is some small loss of performance right at the upper scale. So getting up to near full machine capacity, I guess this is to be expected to a certain extent, where so even though we've got 10 billion lattice sites, the amount of computation that's being distributed to each core is no longer sufficient to outweigh the communication needed to go from all this surround between the different cores. So that's one factor, but there's also the factor that as we're running at full machine scale, you may be potentially hamstrung by nodes that aren't performing for whatever given reason. And so if you have a single node that's not performing well and you're running at full scale, you will be hamstrung by that node. Whereas smaller nodes, you have a better chance of missing nodes that are having a bad day. But otherwise, outside of those factors, we're very happy with this performance curve and demonstrates that HemeLB has excellent scaling characteristics that can be taken up to full machine scale. We've conducted similar cases with the self-coupled test. The self-coupled version of HemeLB, where we have two instances of HemeLB that are running as part of a single job, and these communicate boundary data between them as their coupling scheme dictates. So in this case, we have observed strong scaling up to 96,000 cores, so roughly a third the size of the single instant case, but we've seen good relative efficiency within this case, and we believe this is a very good result given that our self-coupled scheme has not yet been optimized for scaling performance. And so we have also had execution up to 144,000 cores, so almost half machine scale, but didn't quite see the same scaling performance beyond that. So these are things that can be further improved and something we hope to again do with the POP Center of Excellence in the future. An extra point, I guess extra points that come out of this is that being a coupled model, we do expect there to be lesser performance than the single component code, because you have that communication between the codes. We also, as part of generating these results, and in this 96,000 core case, we discovered that we ran the largest known real-world deployment of the Intel vTune profiling tool. So this is, I guess, a demonstration that those tools do work at very large scales and helped us to identify a couple of issues that were occurring in our code at that scale. So as we move towards larger supercomputers and larger compute scales, we are continuing to identify new challenges. And one of those, as I mentioned, where geometries need to be truly massive to occupy machines at this scale, means that we need to generate large geometries. And so this might be multi-tens of billions of lattice sites in our case to fully occupy a machine. And so the good thing is that in order to generate a full human simulation of arteries or veins at sufficient resolution, particularly in very small vessels, this is demanding on the order of multi-tens of billions of lattice sites. However, generating domains of this size is not a trivial task and challenges the memory and wall and allocation limits of current HPC systems, not just SuperMUC, but this is a more broad problem. So working with the team at LRZ, we have been able to develop a refining tool that can take a relatively coarse domain and then improve that to a higher resolution domain, whilst omitting the most computationally intensive steps that can cause us problems along the way. So this has been a good test case, and we've generated a domain with over 38 billion lattice sites that we're using, particularly in our GPU model, as a test case to see how big can we go. I guess new challenges that come up with these new problems is how can we actually launch these domains? And because of how big they are and the memory that they require, this can cause some problems and be a challenge to launch and just execute a simple simulation with these domains of this size. And so these are problems that we are continuing to work on into the future. So now for a bit more case study type problems. So we're looking at arteriovenous fistula. And so for patients with kidney failure, they often need to have their blood cleaned through a dialysis process. And so this is facilitated by an arteriovenous fistula, which is an artificially created joining between an artery and a vein, particularly in the forearm. This increases flow through the vein in particular, and this provides an access point where blood can be removed from the body, cleaned externally, and then returned. And so this provides a nice setting for us to use our self-coupled scheme, where we can do the arteries in one simulation and the veins in the other, and have them linked through a fistula connection. So here are some initial results that we're working on, and these are things that we're constantly improving to develop and improve. But I guess the headline point to look at is the figure on the right, where our initial case study data, ours is the black dot, and it's matching well with correlations and data observations that were observed in clinical observations. So this is a good indication that our work is on the right track and that we are doing the right things with our coupling model. So other things that we need to focus on moving forward. So for example, things like improving various physics aspects of our models, such as including elastic wall effects at our human scales. As I mentioned, geometry generation. There's always performance improvements that could be made in order to get those last few percentage improvements in scaling, particularly at the upper end of machine performance. So for example, furthering improving the load balancing, either through active or passive approaches, and for machines like SuperMUC, taking advantage of specific vectorization instructions. We're also working with LRZ using the Osprey modules to improve visualization and do almost real-time simulation of blood flow simulations. So that's something that we're quite interested to work towards further. So finally, it's just a bit of a summary. We're working towards virtual human simulation using HemeLB to characterize the blood flow at this full scale. And this is a problem that we're looking towards taking towards exascale machines. Achieving and assessing performance on just large scale, but also the heterogeneous architectures requires significant contributions from developers, HPC, and performance analysis experts, all of which working together to get these results. Which HemeLB has demonstrated how this can be done on SuperMUC NG in the context of human scale blood flow to a very good level. And we've demonstrated excellent performance up to full machine scale. We've also demonstrated case study where it can be used in practical application. So thank you very much for listening to my talk. I will stop recording now, and if there's any questions, I will take those live. Thank you.